Kia ora and welcome to No ME, the podcast video series where guest speakers talk about pressing issues surrounding myalgic encephalomyelitis, chronic fatigue syndrome, and long COVID. Join researchers, clinicians, and patient advocates as we take a deep dive into the most recent evidence-based information so that you too can know ME. In today's episode, we will discuss the latest biomedical research conducted by Emeritus Professor Warren Tate. Professor Warren Tate has been an academic researcher at the University of Otago in Dunedin for 55 years. He is a Fellow of the Royal Society of New Zealand, a Fellow of the Alexander von Humboldt Foundation of Germany, and was an International Research Scholar of the Howard Hughes Medical Institute in the States. As a discovery molecular biologist, Professor Tate co-discovered a new rare mechanism of gene regulation utilized by HIV-1. He worked on mammalian memory and Alzheimer's disease and the development of a potential therapeutic agent. Since 2012, Professor Tate has focused a research program on ME/CFS with preclinical studies of New Zealand patients to understand its biological basis. Professor Tate approaches research on ME/CFS through that dual lens of an affected family member of over 30 years and as a biomedical researcher. He has found molecular signatures for the illness, supporting its biological basis, and is now studying how close long COVID is in its molecular signatures to ME/CFS. Good morning, Professor Warren Tate. Thank you very much for joining us here today. I thought we'd just start with finding out a little bit about yourself and your work history and what prompted you to start doing research into ME/CFS, and how long have you been doing that research?
Well, I guess the passion and drive for me to do this started with illness in my own family back in 1990 when my 14-year-old daughter, very vibrant teenager, came down with what was then an unknown illness. Later diagnosed as ME/CFS, and we've lived with that essentially every day ever since, now in excess of 35 years, as she is still in her life. So that drove me initially to try and actually establish a research program to understand the pervasive fatigue that we saw. But at that time, when I put in grant applications, referees said this isn't a real illness, or it's so complex you'd never find out anything useful. So really, it wasn't until 20 years later that I was able to get started on ME/CFS research, and it coincided with the time that the retrovirus XMRV was proposed to be the underlying cause, and so that gave a sort of an impetus. And in fact, I wrote to ANZMES at that time just to see whether they would give me a small initial grant so we could get started. And as we know now, that XMRV was a false lead. It was a false proposition, which was very sad for the ME/CFS community, I think. But I was able to keep going. And so for since really 2012, I've worked intensely on an ME/CFS experimental program, based on New Zealand patients, because I do it from the dual lens of a ME family and also a biomedical researcher.
It's wonderful to have that dedication that you have committed to this cause and finding out and trying to understand ME. And obviously we have long COVID now. What is the current understanding of long COVID when it's compared to ME/CFS?
Well, I was really interested in what the initial reaction was around the world, really, because many clinicians and health professionals were saying, what is this illness? Suddenly people got over their COVID, and then they developed a longer term fatigue, often illness, and many had not seemed to have any knowledge that we already had existing illnesses in the world. I had even similar experiences in New Zealand from quite experienced science people. One person, senior person in the agency that was working on the COVID pandemic in New Zealand, when I had to give a talk and he was in the same program to financial services sector. And the theme was, what's going to be the impact of long COVID on our society? So I said, the first thing I wanted to say, this isn't a new type of illness. We've known about this type of illness. We've had quite a lot of experience about it. And I used that as the basis, and then went to talk what I knew about long COVID. He got up after me and said he has to politely disagree that long COVID is not a new illness. And I didn't want to get involved in the semantics of the argument, but that it just gave an indication how there was a mindset, I think, and people were excited about thinking about this new illness, but not really wanting to acknowledge something that had been more or less under the radar for such a long period of time. And I guess, therefore, for me, this was an important question to help make that linkage, strong, so people would understand we have an illness which can add a lot to our understanding of the new illness of long COVID, but this will be vice versa as well. So I was very grateful then when the World Health Organization in 2021 came out with a clinical case definition, even though I was very critical of the way they did it. They did an iterative process to develop a clinical case definition from people who were affected by it and experts, and took the most dominant symptoms. And the positive of that was it came out looking just like ME/CFS. Even though in their document they didn't mention ME/CFS, there was a reference to a paper to systemic exercise intolerance syndrome, which of course the World Health, the Institute of Medicine in the US actually suggested might be a new name for the illness, but nothing about ME/CFS. But at least the clinical case definition stood up very strongly, and many of the symptoms, of course, overlapped with ME/CFS. The negative about that, it ignored that long COVID was really a heterogeneous illness. We knew that because it contained people that had ongoing organ issues, lungs and heart and kidney and even brain, and so that was, they may not be, but they could be separate from an ME/CFS-like illness. And so a lot of international studies on long COVID, using the Recover money, this $1.15 billion, have sort of worked on cohorts which were heterogeneous, and that's been a slight negative, really. And we desperately needed to have the illness sort of separated, if you like, into clinical phenotypes. And that's just come about now this year, where at least in one what's called an orchestra study from the Netherlands, they've defined out of about 1,800 long COVID patients 4 clinical phenotypes. I'm not sure I totally agree with it, but at least the process has occurred. And what we suspected was that the ME/CFS-like syndrome they listed as 42% of their cohort. In other words, about half, which is what I'd always thought probably it was like. And then they had a respiratory syndrome of 23%, a pain syndrome and neurosensory. Now, we might put the pain syndrome and the neurosensory as part of ME/CFS if we were classifying it there. So it could be as many as 3/4 of the long COVID patients have an ME/CFS-like syndrome, and maybe 1/4 because of the initial lung infection with the virus was very damaging on the lungs. But of course, this also raises the question that ME/CFS as well might have its own clinical phenotypes, and maybe we can, touch on that further later. And so there could be even more than 4. And so this is an important issue to be resolved, to know that, what are we talking about when we talk about these illnesses? And what my own research has shown on individual patients now with longitudinal studies, which I wanted to get onto, just is that there are certainly unique molecular changes in each patient. It ends up with very similar clinical phenotype at the end, but it means that when you're trying to get therapeutics to work on every patient, some may work on some and not on others. And that's, of course, what our experience has been, is that, some patients will benefit from a promising therapeutic approach, whether it be drug or nutraceutical or behavioral kind of therapy. Others will have no effect and some will be harmed by it. So, that's an issue which, with both illnesses, we need to kind of resolve.
Yeah, absolutely. We have known, or at least it's been a theory, hasn't it, that ME has subgroups because it's so highly individualistic that people are fitting into these different groups. It doesn't seem to be dependent on the trigger or anything. It's just how it's affected the body itself and all the systems within the body. Is that right? Yes.
In fact, we've done our comparative, one of the comparative studies with ME and long COVID has been just to look at that question. If long COVID comes from one trigger, the SARS virus, you might expect a smaller range of symptoms, for example. But what you see is almost, identically overlapping with ME/CFS from that single trigger as multiple triggers. So certainly it was a very, plausible proposition that maybe the triggers were really producing the clinical phenotypes, but I certainly don't think that. And I have an example in my University of Otago young women's study group, whereas there's a small group of 5, and each of them have got a different trigger for their illness, and yet what you'd call their clinical phenotype looks almost identical. In other words, they've had the young woman, same age, they've had the disease for about the same period of time, they have the same kind of relative limit of activity, but moderately good for ME/CFS patients in the sense they can come to university, walk around campus, go to a yoga session once a week, but not go out partying and having alcohol or anything, like that. So, here you have what looks like a homogeneous group, but came from different triggers. So I think, your statement at the beginning that it doesn't seem to depend on the trigger is a very accurate one.
And so speaking of your research, you're currently doing research with ME and long COVID. What are you hoping to achieve, or what is the hypothesis that you're actually testing?
So the underlying hypothesis really is to get evidence that long COVID is really a specific example of an ME/CFS-like illness, which of course is now an umbrella term for post-stressor fatigue syndromes that has arisen mainly from viruses, but also from other sources as well. And of course, the earlier name for ME/CFS was Epstein-Barr, post-Epstein-Barr syndrome virus, so rather like long COVID, because everybody initially thought this was coming just from the Epstein-Barr virus, we now know. So wanting to just get evidence that there is a definite link between the two. That's not to say long COVID will be identical to ME/CFS, because there'll be specific features that virus, might bring to affecting the body physiology. Yeah, so that's the underlying hypothesis. I also wanted to do more than just do experimental research, though. The important thing is both policy and education for New Zealand society. And in education, and at the Otago Medical School this year, I gave a lecture specific to that theme. What is the relationship between ME/CFS and long COVID, and what do we have to do for patients to emphasise the patient end of it? And it was good because the Victoria University study for the ministry had come out on the impacts of COVID and had a section on long COVID, and they did a survey just like we've done. I'll talk a little bit about that later. to ask, and part of it asks, what do these long COVID people want from their clinicians? And it was the same as people have been asking for ME, patients have been asking, that is understanding a management plan for how they might, manage their illness, but mainly empathy. So those things were coming through very strongly. So I emphasized that in my lecture. Interestingly, the organisers of the program didn't want me to say that that long COVID was just like ME/CFS, because they knew that I say that 95% of people who get ME/CFS, once they've got their diagnosis, of course, it normally is, most people not until they've had it for a year or two. 95% of people have it for life. They didn't want me to tell medical students this because they thought they'll upset the students, and and I said, well, they're medical students, they're going to be doctors, they have to be told what the situation is. But because long COVID was diagnosed very early in the illness, that is to say, 3 months or even before 3 months it's happening, whereas ME people get diagnosed only when their disease is entrenched, even though the limit is supposedly 6 months, it's often not to a year or two. We can see a difference in a way. What we're looking at, I think, is a window of what probably happens in ME/CFS people when they first get the illness, but it's never been recorded really. If people recover in the first 6 months, then they go away and they don't really think about it anymore. So so education is important really, to establish the link, to realize there are two groups of patients out there in society that need a lot of help, understanding, empathy, and it means that the clinicians, nationally need to be well informed about it, to give, so that any patient anywhere in New Zealand can get the same standard of care. I think that would be one of my goals. Of course, that impinges on policy too, because what I've found is there's been quite a lot of good excitement about trying to do things for long COVID in the ministry, and talking about having national platforms, and I would say national education in medical schools, but less enthusiasm to include another group of people, ME/CFS, an equally large group of people, if you like, onto that. And so at a policy level, what I wanted to achieve is to, just push hard to make sure that connection is made at the policy level, and the ME/CFS community don't get left out. I'm excited that there is this initiative to try and do better for the long COVID community, although time has gone on and we haven't really got too much yet. There was one specialist clinic in Christchurch that was alive for about 6 months, and GPs that managed to get any of their patients into that clinic were very enthusiastic that it was helping the patients, and now it's closed down. So we're not doing very well on the ground right now.
No, and that was because the funding ended.
Absolutely.
And so I think, like you say, with policy, they have to realise the importance of keeping the funding going and actually having a dedicated funding stream to help people, because it's not, 6 months is not enough.
No.
If you think, and I know you've spoken of this before, and perhaps you can elaborate, with it being a virus, and it explains why they didn't want you to talk about long COVID being the same as ME, was because of that mindset of, if it's like ME, then I'm never going to get better.
Right, exactly. Yeah.
Whereas with long COVID, because they're getting diagnosed a lot sooner, they've got that opportunity to put in place all those symptom management techniques, all of the pacing and everything. And they can heal potentially. They've got a more opportunity for recovery. And I think you've said that it takes, for a virus, it takes, a certain period of time fully for the body to come back to homeostasis. I think you've said, what is it, about 18 months?
Well, I mean, it varies from individuals. 50% of people will come back in 2 weeks. Another 20% might take up to 3 months. And, and then there's a fall off, but there'll be some people that take much longer periods of time. And that's true. But what you just, how you summarize that brilliantly, because in a way, that's what I told the leaders of this program that, when in preparation, I told them I was giving a different lecture this year, was just what you said, because I had the opportunity to talk about, there is an opportunity now on what's happening to do things a lot better and get a higher percentage of recovery. And of course, we know now, again, the international figures are all over the place, but there's one study where they claim there were 19% of people with COVID having long COVID at 1 year, and then that had dropped to 11% by 2 years. Now, whether that means they fully recovered or they're just a lot better, I don't know. But, there are opportunities, I think, which can translate to new ME CFS cases as well, which is the great, opportunity.
Absolutely. And your research project includes Auckland-based immunologist Dr. Anna Brooks. So what is it that you're working on together?
So the great thing about Anna is she, when long COVID came, as an immunologist, she, was, and also, from some of her own personal experience, she was quite passionate about doing something for this community. So we, despite our different genders and age, we formed a natural bond, and we've been talking regularly. And so the plan was that she would take bloods from many long COVID patients, and she's building up that base and do cellular immunology on them. And it's a little bit, I don't want to use the word easier, but it's a bit simpler and less expensive to do multiple patients for the cellular immunology than it is for the complex molecular studies that I do. So the idea was that she would get data from all of the patients, and then the interesting, if you like, this is where subgroups come in. If there are subtypes that look as though it would be good to investigate at the molecular level and see exactly what's going on, then I might take 10% of those and do studies on those. So we've got both cellular immunology and molecular immunology alongside it. So that's exciting. As always, in terms of, for us both getting enough funding and whatever, there's that same pressures that everyone's having. So it's been a bit delayed, but the passion is still there to do it, as it were.
Yeah, that's really great. And so you've already touched on how similar that you think that long COVID and ME are. What's the research actually showing us?
Okay.
Yeah.
So, when I started the ME/CFS research, I saw that it wasn't sufficient to do what I would normally do as a biomedical scientist, and that is to study one aspect in absolute depth, because that wasn't going to lead me to understand the illness. All of the experimental studies, in the literature at that time, they were interesting, but they didn't give us an integrated picture. So my philosophy was to try and get a holistic approach of the pathophysiology of ME/CFS from our multi-molecular studies on New Zealand patients. And so we published that first model in 2022, but by then, of course, COVID had come, and so we were able to adapt that to what was known about long COVID, and it wasn't too difficult to form a holistic model that fitted both illnesses, given that we were looking at a kind of an overview of the key features of the illnesses and how those key features is linked up, in other words, the arrows between them. And what I was wanting to do was highlight what are the possible mechanisms that underneath the arrows, and what do they represent as therapeutic targets. So that was the sort of underlying aim of it. And so there's that. And then from that, really a question which I thought had never really been properly addressed is, why do a significant minority of people get these conditions? You know, why do 10% of people with COVID, no matter how serious or mild their illness has been, end up getting long COVID? Why do maybe 5% of people who get glandular fever, Epstein-Barr virus, why do they go on and get ME/CFS, and so on? And so we don't have really a good understanding of that. And in fact, there have been attempts of genetic studies with what's called genome-wide association screens, looking for individual variations at single bases in the DNA, whether they can predict illness. And those have been singularly disappointing. But so what we did in 2022, we wanted to just do a qualitative study with ME patients and long COVID patients, and asking them whether, well, a number of questions, but a couple of critical questions were the family members also suffering from some fatigue-type illness, not necessarily ME/CFS or long COVID. So we managed them as the two separate groups. And what we found is that 28% of people who'd got long COVID had a family member with a fatigue-type illness. So that's quite high. But actually almost half of the ME/CFS people could identify someone in their family. And even another question was, with the long COVIDs, how many family members also got long COVID? And that was about 20%. So a high proportion of people. Now that's suggesting a genetic susceptibility, a familial sort of factor. We also asked, how do they handle childhood illnesses? You know, what length of time do they need to recover? And about 2/3 of the cohorts, and more in the ME/CFS group, said they either had frequent childhood illnesses and or took longer to recover from them. And so instead of the 0 to 2 week period for most of us, they were taking 2 weeks to 2 months. That's that. So we could see also there was something in their personal health history which suggested they had a genetic susceptibility just for, you know, the less serious viral illnesses. The other kind of question we asked was, can you identify anything in your personal life, any environmental, what we call environmental things, so that's the nurture, that might have led you to a predisposition to getting this disease later when the trigger came? And a high proportion of them could. I mean, it might have been glandular fever, which they took a long time to recover from. So that was a kind of a first trigger. They recovered, but it primed them somehow, left them with a mark that when the second trigger came, they got the disease, or some major stressful event of their life, some identified. So a major stressful, I think, essentially primes the person to actually, if they're hit with a second event, they can develop the disease. So we came out of that study thinking there's both genetic factors, definitely genetic susceptibility at this point undetermined, but also environmental effects can push people without that genetic susceptibility into the danger zone. Now, what's happened since December 2022 is that a group in Oxford, a company called Precision, has combined this genome-wide association screen looking for individual variations with a combinatorial platform, you know, huge computing involved, and it allows them to link variations together and to see if they can get combinations which will predict the illness. And they took samples from the UK Biobank of ME/CFS initially. And in fact, they found small clusters of variations linking to just 14 genes could predict 91% of the patients for ME/CFS. Now that's a huge step up, in my view, of understanding the genetic variation. It says now we at least we can see what's on the landscape. We can see what's possible in terms of trying to do interventions. They've now published a preprint of a paper on long COVID and done a comparison of long COVID with ME/CFS as well. And so, and again, there seem to be similarities and differences, but they've got a whole lot more genes that seem to be connected with long COVID. So we'll see where that goes, but that's the first sort of publication of at least actual sites of genetic susceptibility and linking it to particular genes. So I'm, very excited about that, and it's particularly coming on the back of our, more simple qualitative study, which said they've got to be there. Someone's got to find them. So so that's that. Now, experimentally, we've now completed 2 major studies. One that we've just got all of the data and analysis done for was on the epigenetic code, the DNA methylome. And so in that study, we recruited 5 patients of the original long COVID cohort in New Zealand when it first came. And through Ros Vellings, we got 5 ME/CFS patients, which were age gender linked to them, and age gender linked healthy controls. So just 5 patients of each group. And we looked at all of their DNA methylomes, that is what their epigenetic code is doing. And what we've discovered is that 20 genes now that are changed in both long COVID and ME/CFS. So that's like a common methylome change signature. So these are the light switches, the dimmer switches on the DNA, which change the expression of genes. Interestingly enough, when we compare with the healthy controls, those that are where the switches are relatively dim, in all cases in ME/CFS, they're virtually turned off. They're made very dim. And when the switches the dimmers are hardly on at all, in other words, the light's quite bright, they're actually turned on even brighter. So it goes either turning off or turning on. And so a mixture of those 2 things. When we look at long COVID, we find mainly the same trend, but 1 or 2 exceptions, 1 or 2 places where where the dimmer light is not being turned on as it is in ME/CFS, but it's being actually turned off. In other words, the light is getting brighter, just in a couple of cases. So we've so what we've done is what's called whisker plots, where we look at each individual patient. So this is now looking at individual patients rather than groups of patients, as most of the world studies is. And all of our ME/CFS and long COVID patients are separated completely from the healthy controls. Normally in these kind of studies, you see overlaps between controls. They're completely separated. So these events are occurring in all patients. There's a range within the patients, but it's sufficient that it separates from the controls. And that's kind of, quite amazing to me. Definitely.
Yeah, it shows the difference, doesn't it?
Yeah, absolutely. And so then we took so these sites are regulatory elements, but the ones that are best known are promoters for genes, which we know are involved in switching on and off, and also within the gene body itself. So we looked at those. So 9 of the 20 genes were in those, and all of those actually for ME/CFS and long COVID went in the same direction. And remarkably, the degree of change differed from each gene, but between ME and CFS and long COVID patients was virtually the same. So remarkable concordance. I hadn't expected there would be such concordance, and that's kind of, we're talking really about the average then of the 5 patients in each group. So, I mean, that's been quite an exciting study, and we've got good data. And then we said, well, what are these 9 genes doing? And one's ion channel, one's a transcription factor, which affects immune cell proteins. There's a mitochondrial protein involved in that. So the genes that are being affected are genes that we know are important in essentially this switch from the transient immune response to a chronic immune response in those illnesses. So, you know, I think we're pretty excited what's come out of that study.
Can I just say, so basically what's, in layman's terms, what that's saying is that these genes are being switched on or off, and that's creating the repetition, the repeating, relapsing of the disease. So instead of just getting better after a trigger, the genes are actually causing it to remain chronic.
Yeah, that's right, remain chronic. If we talk about a further relapse, which I thought is where you were going, but what we can say, and we've done with our relapse recovery study in ME/CFS, is that we get further changes. And again, that's a remarkable thing when a patient, you know, I've done this relapse study with 2 patients, when they, and both of them had a relapse, when they go into a relapse, their degree of methylation of a particular site month by month is pretty well the same, you know, just as much as we can measure. I think it's probably just our measurement noise. But when it goes into a relapse, there's another change, you know, an additional change. It either goes up or down considerably. So the same phenomenon I talked about before, but an extreme. It's like a second step, you know, absolutely turning it all off or absolutely turning it all on. And so we were able to show in the 2 patients, there were unique sites in each patient, different from one to another, that both turned on or turned off, you know, but at the end of the day, their relapse had the same phenotype. But that it affected their energy production, and we could determine that theoretically from the genes and the pathways that were affected. So it was immune system, it was energy production, it was metabolism, it was gene expression. But each patient had 1 or 2 special things to them, which weren't in the other one. So, you know, we can in the end get the overall effect, which looks like a similar kind of relapse in a patient, but there can still be some unique features of it which aren't necessarily showing out in the dominant neurological symptoms. Okay.
Which can make it difficult for GPs when they have patients, because no one patient is the same as another.
Absolutely.
And it, you know, it can translate as somebody's got really bad gastrointestinal symptoms. The other patient might have all the cognitive impairment symptoms.
Absolutely. I mean, it explains so many things of what we've known about the difficulties of this illness, you know. Okay. So would you like me to talk about the proteome now? Or?
Yeah, absolutely.
So what we did with that, we did long COVID patients and healthy controls. We'd already done an ME/CFS study that we published in 2020. So we used that data and recalculated it all according to exactly the same criteria we'd use for the long COVID study. So the first thing is that we could, in what's called a principal component analysis, that is to say, what is different about these patients versus the healthy controls, and all of the patients in one of the principal components separated from all of the long COVIDs separated from the healthy controls. And this is why you can do such small patient studies. You know, I've been criticized by some of the international people for not doing 1,000 patients, but you know, I tell them
takes money.
Yes, that's right, money, and also other resources in a small country. But,
exactly.
But anyway, so we got that. So then we looked at how many differentially regulated proteins there were, and we have quite strict statistical limits. We got 162 proteins were differentially regulated, some up and some down. 37 were immune system proteins and 21 were mitochondrial proteins. So when we looked at and compared that with our ME-CFS study, analysed in the same way, that was richer. We got about twofold, 360 proteins were differentially regulated, and twofold more immune system proteins and twofold more mitochondrial proteins. And I had to ask the question, well, is that just because we did the studies at different times, although we're using the same instrumentation and facility or not? Some of it might be that, but I think most of it is actually richness of the data in ME patients who have had their illness on average of 16 years, that group we did, whereas the long COVID patients are all between 11 months and 1 year. So stage of the illness might be a thing why we're getting more dysregulation in the long-term ME-CFS patients than we have in the long COVID, but that's speculation. So then we took those differentially regulated proteins and did what we call a string analysis, where we look for clusters, in other words, proteins which link together. And what we found in the long COVID was a big cluster on immune system proteins that, and all sorts of different sub titles like immune cell signaling or cytokine production, or infectious agent responses and things like that, but they're all immune system. And then there were two in gene expression, which relates back really to the methylome. One was on transcription, the factors which actually do all the work in expressing the information in the gene, and the other was in what's called the spliceosome. When RNA is produced off the DNA, it's actually processed up so that the ribosome, which makes the proteins, can actually read the signals. And so those, lots of changes in there as well. And there was a small cluster of mitochondrial proteins. When we do that with the richer ME-CFS proteins, we get two immune system clusters. We get gene expression clusters in the same way, sometimes with different names. We get a mitochondrial cluster, and we get one or two more small clusters, largely because of the richness of the gene. So, it's a bit more complex than the methylome. We can say that certainly there's a very similar response, immune system proteins, energy production, and we can see gene expression. But individually, there's quite a lot of variation, but 20 what we call reactome or KEGG pathways, so that's kind of physiological pathways, are common in the two illnesses. And then they each, the analysis brings out other things that are unique to each of them. So, I think this is still saying that the diseases are very similar. It's possibly speaking to the fact that, one's only 11 months, 12 months old, the other is 16 years old. That's almost certainly a significant factor. And, so long COVID is a bit like an errant teenager, it doesn't quite behave exactly as you would want it to, but it's still got all of the characteristics of what it's going to become like. So that's how
exactly.
I put that. Yeah.
Yeah, I mean, it's in its infancy, isn't it, really? And I suspect if you did a longitudinal study with those long COVID patients in a few years' time, you'll start to see that more dysregulation that you're seeing in ME-CFS.
Yeah, and we might get onto this if we've got time, but that's kind of what I see as the great value of long COVID for all of these post-stressor things. We've got now a huge number of patients coming at the same time. We have got biological samples from them early in their disease, and we're able to follow them, which we haven't been able to do with ME-CFS because it's sort of been, they drip feed and clinicians are struggling, and so we just don't have the resources to do that.
Absolutely. And it shows that your research is essentially proving the point. Physiologically, biomedically, we're seeing this dysregulation or dysfunction in immune system, in the mitochondria, which is the powerhouse for energy production, right? So, and that's translating obviously to all the symptoms, the post-exertional malaise.
Absolutely. And that's kind of when I go back to my model, it's quite gratifying because it does fit with the model, and that's why I thought we had to step back and just get a very high level model of all of the things that were going on so we can then dig down. I'm certainly excited with people like Robert Fair, who looks at a particular small feature and comes up with a hypothesis, which is really exciting creative thinking, and these things need to be tested out. But in some ways, I think we also need to have that big picture that we can actually then, interpret everything on the big picture.
Exactly, and demonstrate how it's actually occurring in the body at a cellular level. And it also explains why some of the things that happen aren't in the standard blood test, because they're happening on such a deep cellular complex level, and we just don't test for those things routinely.
Yeah, and that's been one of the things which has beset this, the whole patient group when they go and have their blood screens, and it says, well, there's nothing particularly wrong with you. There's often just things that are at the margins a bit, but nothing. That's not the patient's fault. It's just that we test so few things in body physiology. We only test the things that we know about because illnesses have come up and we've discovered what is there. So this will hopefully, what we've got to go forward now is add another dimension of tests, really.
So what other research are you working on at the moment?
Okay, so, one exciting thing that I hope to get going on is what I call the Dahlia project. And this is where a physiologist at Otago who was working on glucose regulation extracted some biological molecules from the dahlia flower and showed that they were anti-neuroinflammatory. And in fact, he was able to show what inflammatory pathway they were affecting. And so we got together talking about this because it turns out that the pathway that's affected is one that we've shown is elevated in our ME/CFS transcriptomes. So I wondered, of course, whether this extract would have benefit to ME/CF and long COVID patients. So the extract now has been, a company has taken over the interests, and they've got great dahlia plantations in Canterbury and are making the extract, and it's now been launched as a neuroceutical called Dahlia 4. So in principle, I think even in New Zealand, I think it would be accessible to New Zealand patients. But what we want to do is just do a 3-month study where we look, would be a qualitative thing where we get long COVID and ME/CFS patients to take it for 3 months, and we can see whether there's any rapid improvement or delayed improvement by the 3-month, or if they do get rapid improvement, is it gone by 3 months? That's the kind of preliminary study we want to do, just to see whether this has got promise. And the company has agreed to donate us the, it will be about $20,000 worth of the capsules. At the moment, it's stalled in the ethics approval side, largely because I'm so busy that I'm trying to push it along as quickly as possible. But we certainly hope to get that going. I would hope we can get ethics approval by the end of the year, and we can recruit patients and just see, because this might be, we know low-dose naltrexone, which is anti-neuroinflammatory, helps some patients. This could be an alternative anti-neuroinflammatory, which, might just actually be amazingly effective with glucose regulation. What the physiologist has shown in animal models, it actually has almost instant reversal. In other words, they've got a zebrafish model and also a mouse model I've had, and they've shown that you get rapid effects. That's why we want to see whether if it could be something that could rapidly reverse symptoms. That would be
That's
just
very promising.
Yeah, I'm not quite so hopeful about that because humans are humans. They did do a limited clinical trial with this on diabetics and pre-diabetics, and there were promising results. It wasn't quite as spectacular as the zebrafish, which they've got a coloured marker, a fluorescent marker, and the fluorescent marker just completely changes, sort of thing. So, but it was still promising with the humans but they want to just market it as a nutraceutical and not go through all of the pharmaceutical industry controls things to do with inflammation. So if you've got something like this, it's almost impossible to get it takes years and squillions of money to get it approved. So by going through a neuroceutical and calling it something which affects glucose regulation, they don't have to do that. But if that's helpful to ME/CFS patients, there's something that people could buy just out of pharmacies. So I'm so excited about that. So, of course, as you know, we've talked about the vaccine effects from the COVID vaccine that ME/CFS patients suffered at high frequency from ANZMES survey, which I think was, an excellent initiative. And certainly we'd like to do another survey, as we've talked about with ANZMES, just to see where those people are now. Interesting enough, subsequently, I've been contacted by legal firms who are representing patients just who were healthy in the community that, 48 hours after getting a vaccine has been seriously affected and got essentially an ME/CFS-like illness. All of their symptoms are the same, and of course turned down by accident compensation and even insurance companies and things like that. So I've been writing briefs now to explain why I think that these are legitimate claims, both all of the symptomology, the fact that it came at a certain, short time after the vaccine, and it was just out of the blue kind of thing. So, the vaccine effects, and they're also being recognised now more around the world, and that's not to say, wasn't it fantastic that we had a vaccine that many people could take and not get anything, but I think
particularly for the Delta strain, right? That's what it was developed for.
Absolutely,
it definitely reduced hospitalisation and death for
absolutely.
The Delta strain.
And for people like me, and dinosaur age, it's really important to have that protection. And at the university, there's cases, on my floor every week carrying still on. So we need protections. But for
it would be really interesting. Sorry, it would be really interesting to retrospectively speak to those people who've had that harm, who were previously healthy, and look back at their childhood, do that qualitative study, and see. Yeah, is there family members who have some kind of fatigue illness and
that's right. So
childhood susceptibility.
My plan was the person who did our original study, on the genetic susceptibility, who's now working in Sydney, but not with a part-time job. She's an ME/CFS person. She's keen to be involved in this, and she's got the skills now, so we could do that. And we've got, we've now got 2 surveys, one for the Dahlia project she's developed and our original survey, so we could adapt that survey, and I think it could be done quite efficiently. So I hope that my time and my position here is affirmed such that we can go ahead and do that in the future. And I think the important thing is to get general acceptance from the health policy and professional people that we need to we know more about vaccine effects and being able to help people by getting this information. We're not, not trying to undermine vaccine efforts. I mean, vaccines have been fantastic, we didn't know about genetic disease until vaccines came in, and then we suddenly discovered that they were saving people who subsequently, were shown to have genetic disease. So
really absolutely. Oh, yeah. I mean, it's been a huge coup for the medical world to have vaccination. I mean, look what it did for polio. Practically eradicated it. Right. Absolutely. And we're not saying that. Definitely not anti-vax. It's just that unfortunately, for some people, they had a susceptibility. I mean, I had a severe adverse reaction to the second dose, and it changed my baseline functionality so much. I'm still not right. No. A couple of years later. So you just don't know how you're going to react.
Yeah, the first family contact I got was a husband who rang me up about his wife, and he said, my wife's never been this ill with her ME/CFS before, and he was traumatized because just by what it had done. So, that really kind of made me realize how serious some people were being affected, So, yeah, no, all important. So anyway, we want to do that. And then, although, my attempt to get an HRC grant for a diagnostic test based on the methylome was turned down, the pathology collaborator is still very keen to work on this, because we've published 2 papers together on the methylome and ME/CFS, and they've been received extremely well. So we've recruited an Indian student, a very bright Indian student, who will likely work on this. And so the good thing about that, in terms of just succession, if you like, planning to have a major, he's a younger researcher, interested and wanting to carry on sort of working on this, and some quite a lot of the resources will be supplied by him, and I'll be involved, even if I were not to have an experimental group. By then I could still be involved and supervise the student and whatever. So that's kind of just putting in good planning to make sure we can carry on with that.
Absolutely. So I mean, we have touched on it a little bit, but what do you think are the benefits to the ME community and the long COVID community of all this research that's going on, and particularly your own research?
Okay, so one of the things that I think would be fantastic, for families and for patients who have potential to get this disease, is be able to predict those that are susceptible and at risk when a trigger comes. And, I was really impressed with a study by Leonard Jason, who's a kind of a golden oldie in the ME/CFS field, where a paper published in 2021, a follow-up in 2022, where they took blood from 5,000 Northwestern University students in the United States. And of those, 238, it was a 5-year study, got glandular fever. And of those, 55 came down with ME/CFS. And so they were able to go back to the original blood samples and say what was different from those 55 than the others of the 238 that didn't actually come down with a post-viral illness. And it turned out that there are some things like the irritable bowel syndrome, kind of gastrointestinal things, where people had that in the original group that ended up coming down with ME/CFS. There were some things in terms of cytokine metabolism were highlighted. I'm not sure that the study has been very detailed sort of look at them, but at least in the look they've had at those people, there were things there that were potential biomarkers for susceptibility. So I would say, from a patient point of view, if we could determine at-risk families, and our qualitative survey said every family of the people that we recruited, were an at-risk family because they had such connections of other family members, then there could be possibility these people could be tested and prophylactic sort of things put in place if they got struck by a trigger. And just like happened with the vaccination, I mean, Ros Bellings was, giving her patients antihistamine courses and having steroids and ready. And I know Nancy Klimas in the United States did the same, just as a kind of a protective measure to try and help those people if they didn't get a post-vaccine effect. So I think there's huge potentials for that. I think one of the things we've touched on several times is subgroups and clinical phenotypes. I think if we could understand from the long COVID work, because we've got so many patients and they're all kind of same time, we could understand the clinical phenotypes better in each condition and then link them to possible targeting therapeutics. We might be able to then, instead of having every patient trying everything under the sun to improve their position, which is financially limiting, but also it's just such an ad hoc approach, we might be able to have clinicians with enough experience to be able to, push people towards a small number of targeted therapeutics, which is more suitable for them, and not try a lot of other things which clearly, are either not going to affect them or make them worse, as it were. So that I would hope that we could do that. And one of the things is, there are a lot of antioxidants available, like coenzyme Q and MitoQ and some more, and inositol cysteine, and more sort of sophisticated ones. And it does look as though that individual antioxidants might actually operate differently in different patients as well. So having trials of antioxidants so that, one can choose the right antioxidant for the patient, I think would be a really good thing. And of course, I've talked about the neuroinflammatory options. If we get more than low dose naltrexone, we get something like the dahlia extract, and there could be others, that people could try. And we need really, for those, the antioxidant, we need to have some clinical trials. So given that we've got so many long COVID patients and the money's there, we could have proper clinical trials for some of these things. And the other interesting thing to me is that, and also perhaps probiotics for dysbiosis, it's quite clear that the gut immune brain axis is really important. It's not known really yet whether the patient has dysbiosis before as a susceptibility factor, or really the dysbiosis comes after getting the illness. So that has to be properly defined. But you could imagine that there could be a series of probiotics that might help patients restore certain bacteria, which will help them. What interests me too is just hearing about, the papers being published that even for MECFS from people saying that this is a disease you can recover from. When you read the paper, it's not quite so compelling, but the idea, of course, I have the idea. I think it is a condition that you can recover from if we just knew all of the right things. So being able to study long COVID and the patients that recover, and if we've got samples from them to understand why they recovered, what things were they doing, we'll start being able to build up, I think, a picture of how ME/CFS patients and long COVID patients might be able to facilitate their recovery, so we do have a much higher percentage of people recovering than we do now. Obviously, from my own personal, family experience and talking to so many patients, many people are very good at systematically going through a whole lot of things and finding things which benefit them and actually living with those. That's not quite recovery, it's a better management. But it's that kind of, patients will cooperate, I think, if there are good options put in front of them to try. And so, that would be my hope for the future.
Absolutely. Nobody wants to be unwell, especially not with these conditions. So we're all willing to try anything, it seems. Well, thank you so much, Professor Tate. It's been wonderful to have you here. And it sounds like there's lots of invaluable research going on that you're instrumental in making happen, particularly here in New Zealand. It's so important because we have, at least 45,000 people in New Zealand affected. We conservatively estimate it, don't we, at about up to 25,000, but I think it's significantly more based on overseas data. So,
and the other thing I think is that, like last week we had 4,000 cases of COVID in New Zealand. Now, who knows what the percentage will go and get long COVID. It might be 5%, but that's still a significant number each week of new long COVID cases appearing, So when I hear people say, oh, it's all over and we don't need any public health measures and things like this, I internally hemorrhage because it's still, throwing up new cases of what is, a very debilitating illness. As you say, no one wants to be sick for the, a good part of their
lives. Yeah, exactly. So thank you very much. And it sounds like exciting times too, which I think a patient group and hopefully our health professionals too will find exciting, that there is this potential for therapeutics, that we're getting closer, inching bit by bit. Towards some therapeutics. So it is wonderful. And I'd also like to actually say to you, on behalf of the ME community and ANZMES as well, extend our heartfelt thanks to you, because you have dedicated so many years to this and you're still going. You could be retired, but here you are doing the research, but also speaking with media and speaking, here with ourselves, talking to health professionals, talking to the medical students. You do so many hours and it doesn't go unrecognized. We know how valuable it is and we are so very, very grateful to you. So thank you very much for that.
Well, it's a privilege and a pleasure for me to work on behalf of the community, and I've interacted with so many people affected, and I have enormous respect for them and what they've gone through and their struggles. So it's not hard.
I understand. And if anybody would like to contribute to Professor Tate's research, you can do so through the ANZMES link, which is www.anzmes.org.nz. Thank you very much, Warren. Appreciate speaking with you today.
Thank you.